Hello again, Jared Pitts here, and today we're going to be reading another article by Dr. Ray Peat. This one is called Fatigue, Aging, and Recuperation, and you can find it on his website, raypeat.com. Before I get into the bulk of this article, just wanted to remind you I have a new website up called MissouriHealthCoach.com, and you can get in contact with me there or at MissouriHealthCoach@gmail.com. And you can set up a free 15-minute consultation on that website with me, and then we can see how I can help you with your health journey there. I also want to remind you that I have several links to a couple of companies that I trust and use who sell things like aspirin, vitamin K2, vitamin D3, and the likes, and other things that Dr. Peat discussed and suggested. Okay, let's get into the article. I'm excited to read this article because fatigue, aging, recuperation, right? This is kind of a coverall for lack of metabolic function. So you're going to get a lot of what Dr. Peat has researched and has shared with us in this one article. All right, this is how it goes. Old people and sick people tire easily. Surprisingly, little is known to explain that common fact. Myths about lactic acid and oxygen debt have misdirected most fatigue research. The cellular processes involved in fatigue overlap with those of aging. Knowledge about the mechanisms of fatigue should be useful in preventing some tissue swelling disorders, organ failure, degenerative calcification, and other energy-related problems. Okay, so we have a glossary of several words here that I'm going to go through, the word, the definition, and then we're going to get into the main bulk of the article. The first word we have here is uncoupling. In cellular respiration, oxidation of quote-unquote fuel in the mitochondrion is coupled to the phosphorylation of ADP, forming ATP. Uncouplers are chemicals that allow oxidation to proceed without producing the usual amount of ATP. Our next word is DNP, or dinitrophenol. This is an uncoupler that was once popular as a weight loss drug. If you know anything about weight loss drugs, they tend to come out several years later with some kind of lawsuit. I'm not sure about this particular one, but there's one specifically in the mainstream news right now that a lot of people are talking about and might not be the best route to go about losing weight. Our next words are NAD plus and NADH. Nicotinamide adenine dinucleotide. All right, so you can see that NAD in there. And its reduced form are coenzymes for many oxidation and reduction reactions in cells. And in regards to these two molecules, you can kind of think of NAD plus as, and Georgi Dinkoff has explained them in this way, and it's been helpful for me. So NAD plus being like a taxi that doesn't have any riders in it, right? And then NADH has picked up riders, and the riders are electrons, okay? And so it's carrying them along what you would call the electron transport chain, okay? So a lot of the time you can have blockages, meaning you have a bunch of taxi drivers that have their cars filled. So you have no free taxi drivers or NAD plus molecules able to pick up these electrons to carry them across the electron transport chain in order to create energy, right? So you want to be able to free up the NADH into NAD+, if that makes sense. The next word is hyperammonemia, and this is the presence of too much ammonia in the blood. Our next word is Viscinal water. Not entirely sure if I'm saying that 100% correctly. Water near surfaces, especially hydrophobic surfaces, that is physically and chemically different from ordinary water. And next we have hydrophobic, insoluble in water, a nonpolar oil-like molecule that repels water. Okay, so if it's hydrophobic, scared of water is essentially what you would read that as, right? So it can't dissolve in water. Something like a fat, right? You pour olive oil into water, obviously it's hydrophobic, separates out. All right, here we go, the main bulk of this article. Unlike the somewhat technical medical concept of quote-unquote stress, the idea of fatigue is something everyone understands, to some extent. Hans Selye, and if you know anything about Peat, Hans Selye will always be brought up if stress is discussed, okay? Because he did a ton of research into this concept of stress. Okay, Hans Selye's studies of stress weren't widely accepted until about 40 years after their publication. But some of the main investigators of the fatigue phenomenon are still practically unknown in the universities, many years after they published their work. Several things have kept fatigue research from advancing, including the common feeling that fatigue is already sufficiently understood, and that it is somehow trivial compared to problems such as growth, reproduction, and disease. Fatigue is usually described as decreased responsiveness resulting from overexertion. For example, a muscle's decreased strength or speed of contraction, or a nerve's decreased speed of conduction, or a sense organ's decreased ability to detect or to discriminate. Another meaning of fatigue, a decreased resistance or strength, can be applied to materials, as well as to some biological functions, for example, when fatigue leads to sickness or infections. Quote-unquote responsiveness implies sensitivity, and decreased sensitivity to stimulation can be seen in fatigued sense organs, nerves, muscles, and many other types of cell, immune cells, secretory cells, etc. Even plant cells have very similar processes of excitability that can be depleted by repetition. In a series of lectures to the Royal Society in England from 1895 to 1901, the physicist Jagadesh Chandra Bose described work that at first excited and then disturbed many physicists and biologists. He had invented devices for both producing and detecting electromagnetic waves, and he had been the first to produce millimeter-length radio waves, or microwaves. In Marconi's first transatlantic radio transmission, Bose's signal detecting device was used, and you'll recognize that name, Bose, as being, the company that produces speakers and whatnot. This device was based on the fact that two pieces of metal in superficial contact became electrically fused, or cohered, in the presence of an electrical or electromagnetic field. After they cohered, a mechanical shock would separate them, breaking the electrical fusion. When Bose was experimenting with his quote-unquote self-restoring coherer, a semiconducting device that spontaneously broke the connection without being mechanically shaken, he observed that it became insensitive after prolonged use. That is, it lost its self-restoring capacity, but that after a rest it recovered its sensitivity. He recognized the complex behavior of this instrument as being very similar to the electrical physiology of living cells. He then began a series of experiments on plants, animals, and minerals that showed similar responses to all kinds of stimulation, including mechanical, and thermal, and electromagnetic. The idea of metal fatigue wasn't new, but Bose was able to think far beyond the ideas of the metallurgists. Biologists were thinking of electrical responsiveness as a defining feature of life, and Bose demonstrated that plants had electrical responsiveness, very similar to that of animals, but also that similar reactions could be demonstrated in minerals. This was what disturbed the English scientists. Sensitivity, irritability, fatigue, and memory were supposed to be special properties related to life, and maybe to consciousness. For the Englishmen, there were religious implications in this Hindu's research. There were several reasons that European and American scientists couldn't accept the universal nature of the electrical properties that they were studying in animals. One of their motives was to see life as something immaterial, or of an absolutely different nature than inorganic matter. Another problem had to do with the developing belief that the special properties of life were enclosed in the hereditary substance of each cell, and that the electrical functions of cells were produced entirely by the presence of a membrane surrounding a drop of water containing randomly moving dissolved chemicals. For the membrane electricity theory, it was essential to believe in the random behavior of things dissolved in the cell water. So they considered the electrical mechanical reactions and interactions of minerals to be so unlike the processes of life that it was inappropriate to see analogies between them. Minerals were composed of atoms, and according to the doctrine of the time, they could have no quote-unquote physiological functions except in the atomic scale. It was more than 20 years before mainstream physicists began thinking about quote-unquote delocalized forces and fields in minerals. Okay, so we're getting a lot of build-up here. Stick with us. This is the beauty of Dr. Peat's research, in that he goes all the way back. He shows us the narrative of how these ideas came into being and helps us understand the misconceptions and why doctors today believe in a certain way, why everything is so dogmatic, right? When it doesn't have to be dogmatic. So, between 1915 and 1934, Michael Polanyi made many observations that made it clear that the old kind of electrical atomism was completely unfounded. The behavior of mineral crystals and the interactions between different phases of material, such as gas or liquid with a solid, could be understood only in terms of relatively long-range forces. Polanyi's experiments showed, for example, that events on the surface of a crystal modified the strength and deformability of the crystal. Many others between 1900 and 1940, Lepeshkin, Nasanov, Bugenberg, de Jong, and Solko-Tromp, for example, argued that the sensitivity of protoplasm had to be understood in terms of long-range order, something like a liquid crystalline state of matter that would require some of the kinds of knowledge of matter that were being developed by physicists, metallurgists, and a variety of others investigating the condensed states of matter. But the mainstream biologists preferred to describe cells in terms that would make impossible any of the responsivities or sensitivities seen in the quote-unquote simple, solid state of minerals. To defend their ideology of the immateriality of life, they denied that the subtlest features of matter had anything to do with life, reducing life to a debased set of special, merely theoretical mechanisms. The now defunct physical theory of merely localized atomic electrical forces became the paradigm for the new biology. The many demonstrations of coherent, ordered physical behavior of the cytoplasm, for example, Gurwich's mitogenic radiation, were dismissed with prejudice. During G.W. Cryle's long career from 1889 to 1941, understanding shock, biological energy, and fatigue were his main concerns. He believed that shock was the result of brain exhaustion, and in one of his last publications he showed that the brains from exhausted animals produced less bioluminescence than those from rested animals. His importance was in demonstrating that fatigue and shock are systemic conditions of the organism, rather than isolated events in muscles and nerves. Recent publications are showing the validity of this view. Cryle's approach to the prevention and treatment of shock was based on isolating the damaged area with local anesthetics. Blocking the nerves from one injured part of the body, for example, the sciatic nerve in the leg, could preserve energy production and normal cell functions throughout the rest of the body. And I hope he gets more into this, because you'll see this come up a lot in Dr. Peat's work, the use of local anesthetics. About 30 years earlier, in 1901, Vidensky had demonstrated that some types of fatigue appear to be a defensive blocking of responsiveness, such that intense stimulation would produce no response, while weak stimulation could sometimes produce a response. These changes affected cell functions in a variety of ways that he called narcosis and parabiosis. There have been two popular ways to quote-unquote explain fatigue. One by saying that the cell's energy usually thought of as ATP or glycogen, is used up. The other saying that the accumulation of a metabolic product, usually lactic acid, prevents further functioning. The obvious problem with these explanations is that the fatigue response is quite independent of those metabolic changes. Another problem is that those ideas don't explain the real changes that occur in cells that are demonstrating fatigue. Fatigued cells take up water and become heavier. They also become more permeable and leak. When more oxygen is made available, they are less resistant to fatigue. And when the organism is made slightly hypoxic, as at high altitude, muscles have more endurance and are stronger, and nerves conduct more quickly. These facts don't fit with the standard model of the cell, in which its sensitivity is strictly governed by the behavior of its quote-unquote membrane. For example, how can a membrane leak large molecules at the same time that it is intact and causing the cell to swell osmotically? They are consistent with the model of the cell that treats protoplasm as a special phase of matter. Right, so in grade school we learn about the quote-unquote 3 phases of matter, and then they'll mention things like lightning or plasma, but they'll talk about gas, liquid, and solid, and how it can go through those different phases. Okay, and so Dr. Peat talks a lot about this structured water, this special crystalline type structured water phase. Okay. And he also talks a lot about structure and energy and how these are intertwined and they can't be disconnected, right? So I'm sure he'll get into some of that. Another feature of fatigue, and often of aging, stress, and sickness, is that the relaxation of muscles is retarded and impaired. Okay, and so he also talks about this idea of the ability of a cell to relax. This is important in our body, right? Just like our heart, and when it's pumping, it has to pump strongly, but then it also has to relax well. And that doesn't happen when you're not creating energy well, when you have an impaired metabolism, okay? Hypothyroidism causes muscle relaxation to be slowed. Okay, and he'll probably get into this, but I love doing the test on people's Achilles tendon where, you can even use a butter knife for this, but you basically, face a couch and you kneel on it with one of your legs so that, your foot's hanging off the back of it. And maybe I'll post a video to this in the comments, but you can kind of hit the Achilles tendon with a butter knife or with one of those reflex hammers that a doctor might have. And you can watch your foot kind of tense and then relax again, just like when you hit your knee and your leg kicks a little bit. Well, your foot's ability to relax, when you are hypothyroid, you'll watch someone's foot just slowly come back to its original position, okay? Because there is such metabolic impairment that those cells can't relax. They can't create the energy that is required in order to relax, which it kind of like goes against, like if you don't understand this stuff that Dr. Peat's talking about, then that, that wouldn't make sense. Why would something have trouble relaxing? Okay, but that's what he's going to get into here. All right, so hypothyroidism causes muscle relaxation to be slowed, both in skeletal muscles and in the heart. F.Z. Mearson showed that stress causes heart muscles to be exposed to increased calcium, followed by breakdown of fats and proteins, and that these changes keep the injured heart in a continuous state of partial contraction, making it stiff, and resistant to complete contractile shortening. When many cardiologists talk about the heart's stiffness, they are thinking of muscular thickening and fibrosis. But those are late consequences of the kind of contractile, unrelaxed stiffness that Mearson described. The hypothyroid heart does eventually become fibrotic, but before that it is just unable to relax properly and unable to contract fully. This failure to empty fully with each contraction is a kind of quote-unquote heart failure. But it can be corrected very quickly by supplementing thyroid. Even the fibrotic heart can recover under the influence of adequate thyroid. The analogy of the quote-unquote coherer would suggest that the overstimulated muscle isn't able to decohere itself until it has had a rest. It responds to stimulation, lets the energy flow, but then can't turn it off, and the energy keeps flowing because of a change in physical state. Albert Szent-Györgyi was probably the first person to seriously investigate the semiconducting properties of living material, since he was aware of W.F. Koch's idea of a free radical catalyst to support oxidative metabolism. His suggestion in 1941 that cellular proteins could function as electrical conductors, or semiconductors, was very likely based on his research in cellular respiration, as well as on his work with muscle proteins. He had observed that ATP lowers the viscosity of a solution of the muscle protein myosin, and that it would cause a filament formed by precipitating myosin to contract. The polymerization, and polymerization, that's kind of like combining things into one, right? You hear kind of the word polymer in there, like a plastic, how plastics are like these long carbon chains. The polymerization and contraction of proteins under the influence of free radicals was at the heart of F.W. Koch's therapeutic ideas, but Koch's work was about 100 years too early by medical standards, as most good ideas are. Szent-Györgyi observed that although ATP was involved in the contraction of muscles, its post-mortem disappearance caused the contraction and hardening of muscles known as rigor mortis. When he put hardened dead muscles into a solution of ATP, they relaxed and softened. The relaxed state is a state with adequate energy reserves. After Szent-Györgyi moved to the U.S. in 1947, he demonstrated the effect of muscle cytoplasm on the behavior of fluorescent substances, which was analogous to that of ice, until the muscle was stimulated. During contraction, the fluorescent material behaved as it would in ordinary liquid water. This effect involved the stabilization of the excited state of electrons. This single demonstration should have caused biologists to abandon the membrane theory of cellular excitation and to return to basic physics for their understanding of cell behavior. The implications of Szent-Györgyi's work were enormous for biology and medicine, and even for the understanding of semiconductors. But most of the world was hypnotized by a simple textbook model of cell membranes. Szent-Györgyi also demonstrated that the combination of properly balanced electron donors and electron acceptors, or Da pairs, and I talked about this at the beginning with NAD+ and NADH, would cause a muscle to contract. He compared this to quote-unquote doping an inorganic superconductor to regulate its electronic behavior. Although these experiments were done half a century after Koch's application of free radical chemistry to medicine, they still didn't rouse the pharmaceutical industry from its toxic slumber. I suspect that it was Szent-Györgyi's research with those interesting electronic properties of cellular water and proteins that in 1960 gave Linus Pauling the idea to explain anesthesia, specifically noble gas anesthesia, in terms of water clathrate formation, or clathrate formation. The restructuring of cellular water by the hydrophobic atom or molecule of an anesthetic. His suggestion caused a reaction among biologists that discouraged research into the subject for about 40 years. Gilbert Ling, and you'll find his name anytime Dr. Peat talks about cell membranes, right? So Gilbert Ling did a lot of research and writing in order to debunk this idea of cell membranes, essentially. Gilbert Ling's view of cytoplasmic structure gives a different emphasis to the function of electrons, which I think is an essential complement to Szent-Györgyi's view. Ling's emphasis is on how the inductive effect of adsorbed substances, for example, ATP and progesterone, has powerful adsorptive effects on proteins. Okay, let me read this again. Ling's emphasis is on how the inductive effect of adsorbed substances on proteins changes the charge concentration on ionizable groups. When the charge concentration is in one configuration, more acidic, The preferred counterion is potassium, and in another, less acidic configuration, it is sodium. Gilbert Ling's biophysical calculations were useful to physical chemists, and were soon put to practical use for understanding ion exchange resins, such as water softeners. Many sorts of evidence showed their validity for cell physiology, but nearly all biologists rejected them, preferring to talk about membranes, pumps, and channels, despite the evidence showing that the properties ascribed to those are simply impossible. NMR imaging, or MRI, was developed by Raymond Damadian, specifically as an application of Ling's description of cell physiology. Although metals are conductors, the function of the coherers of Bose and others shows that the surface is a semiconductor that requires the slight excitation of an electromagnetic wave to become conductive, at which point the conductive band of electrons in the metal becomes coherent and extends from one particle into the others. The surface of any phase of a substance has electronic properties distinct from those of the bulk phase, and in a sense, the interface constitutes a special phase of matter. When the electrons of the interface lose their special properties, the structure of the whole system changes. Okay, so just think about this all in terms of a cell, right? Talking about this, the surface of the cell. Is it a membrane, quote unquote, in how, mainstream medicine thinks of it? Or is it a special phase of substance, right, that just has different electrical properties, distinct from maybe the inside of the cell, right? When a muscle cell is stimulated enough to cause a contraction, the interruption of its resting phase causes a shift in the charge concentration on the proteins. Potassium ions are exchanged for sodium ions. Calcium ions enter and phosphate ions separate from ATP and are replaced by the transfer of phosphate to ADP from creatine phosphate. Since the quantum physicist E. Schrödinger wrote his book, Time's Arrow, people have often thought of life in terms of negentropy, going against the general tendency of entropy to increase, except for aging and death, which are seen as obeying a law of increasing entropy. But A. Zoten investigated organisms rather than abstractions about electrons, and shows that aging involves a decrease in entropy and a slowing of metabolism. The decrease of entropy with aging, according to his view, would be analogous to crystallization, a sort of progressive freezing. When a nerve is stimulated, it releases energy suddenly, and much of this heat seems to be the result of a change of structure in the cytoplasm, since during the resting recovery of the nerve, its temperature goes slightly below the ambient temperature, despite the release of some heat from the chemical changes of metabolism stimulated by the nerve's activity. When a physical change is endothermic, as the nerve's recovery is, that can be interpreted as an increase in overall entropy, as when a rubber band spontaneously contracts and becomes cooler. Okay? So, just quick explanation here for those that don't really have a background in chemistry. You have different reactions in chemistry, right? You can think of one of the most basic reactions of like vinegar and baking soda, right? This is a chemical reaction. Two things interact, right? And they form new things. And some of those products can be heat, right? The product of a reaction could be heat, one of the products. Now you can have an exothermic reaction where you feel heat around this reaction. Well, why would you feel heat? And the reason being is heat is being released, okay? And so maybe from the bonds that are being broken between atoms, right? Whereas an endothermic reaction is when it feels cold around a particular reaction, okay? That's because heat is being absorbed, essentially, into these bonds that are being created. Okay, it's pulling heat or energy out of an environment. Okay, so I hope that helps. Bose's rested coherer, which with time spontaneously recovered its semiconductive, or relatively insulating, property wasn't being powered by metabolism. As the particles returned to the relatively isolated state, there was a decrease of order. And the change was probably somewhat like the spontaneous energy change in the stimulated crustacean nerve. I assumed the change would result from the absorption of environmental heat, possibly with infrared resonance with electron conduction bands. Seeing the structure of cytoplasm as something like a spring-driven mechanism, able to bounce between two states or quote-unquote phases, makes it easier to see cellular fatigue as something different from the various metabolic energy sources, ATP, glycogen, and oxygen, which, contrary to conventional assumptions, aren't closely tied to the functional losses occurring in fatigue. The role of metabolism then becomes analogous to the role of the quote-unquote tapper in the early forms of the coherer. Water in its normal state is a dielectric, but when it is polarized by an electrical charge or by the presence of a phase boundary, its normal state is altered. This is the special interfacial water or Viscinal water. With the movement of ions, mainly potassium, sodium, calcium, and magnesium, all right, you'll recognize our standard electrolytes. During excitation, the state of the cellular water is necessarily changed by the presence of different substances. In the excited state, cell water is less hydrophobic, more hydrophilic, right? It loves water, than in the relaxed state. A network of quote-unquote hydrophobic interactions extends through the relaxed cell. One of the properties of a dielectric is that it tends to move into the space between charges, with a force similar in principle to that involved in dielectrophoresis. In the resting state, potassium is the main inorganic ion, and it is associated with acidic groups, such as aspartic and glutamic acid. During excitation, potassium is partly exchanged for sodium, which becomes the preferred counterion for the acid groups, and calcium enters the cell along with the sodium. Potassium's interaction with water is very weak. Its hydration has been called negative, allowing water to form the structures that are stable in the presence of hydrophobic surfaces. Sodium, and especially calcium, smaller atoms with higher surface charge concentration, powerfully interact with water molecules more strongly than water interacts with itself, disrupting the delicate, somewhat hydrophobic structures of the intracellular water. Calcium, with its two charges, has important binding and stabilizing functions in the resting cell. In the excited cell, these internal calcium ions are released while extracellular calcium ions enter the cell. With the increased movement of charged particles during the stimulation of a nerve or muscle, as one kind of counterion is exchanged for another, and the destruction of some of the water's structure, there are some opportunities for bulk dielectric water to enter cells, interfering with the arrangement of proteins, and tending to cause swelling and separation of the structural elements of the cell. Electron micrographs of fatigued muscle show a remarkable separation of the actin and myosin proteins. In the excited state, NMR studies showed that cell water behaves more like bulk water. That is, its molecular movements are relatively free, indicating the momentary loss of the interfacial state. In this state, the uptake of water and the fatigue-related swelling of nerve and cells would be driven at least partly by the principle that a dielectric tends to be pulled into the spaces separating charges. The bulk water that enters a cell during the breakdown of viscinal water functions as an extraneous material, somewhat beyond the cell's control. These bulk-like high dielectric properties of water in the excited cellular state can explain many changes of enzyme activity. Previously nonpolar lipids would develop a negative surface charge from accumulating hydroxyl groups, and there's a study, which would tend to increase their oxidation and degradation. With the loss of the interfacial water, the cell's high-energy resting state is replaced by an active mobilization of its resources to maintain and restore the cell's structure. metabolic energy begins to flow into the processes of restoration, serving the function of the tapper in the earliest coherers. Looking at fatigability, muscle contraction, and nerve conduction in a variety of situations, we can test some of the traditional explanations and see how well the newer quote-unquote bioelectronic explanation fits the facts. Osmotic pressure, hydrostatic pressure, atmospheric pressure, and the degree of metabolic stimulation by thyroid hormone affect fatigue in ways that aren't consistent with the membrane electrical doctrine. The production of lactic acid during intense muscle activity led some people to suggest that fatigue occurred when the muscle wasn't getting enough oxygen. But experiments show that fatigue sets in while adequate oxygen is being delivered to the muscle. Underwater divers sometimes get an excess of oxygen, and that often causes muscle fatigue and soreness. At high altitudes, when there is relatively little oxygen, strength and endurance can increase. An excess of oxygen can slow nerve conduction, while hypoxia can accelerate it. Increasing the delivery of oxygen at higher pressure doesn't increase the cellular use of oxygen, or decrease lactic acid production in the exercising muscle. And that's from a study from 2000. But it will increase lipid peroxidation. High hydrostatic pressure causes muscles to contract, though for many years the membrane doctrinaires couldn't accept that. Underwater divers experience brain excitation under very high pressure. Since Viscinal water has a larger volume than ordinary water, analogous to the expansion when ice is formed, though the volume increase in cell water is slightly less, about 4%, than in ice, which is 11% more voluminous than liquid water, compression under high pressure converts Viscinal cell water to the state that occurs in the excited cell, the way ice melts under pressure. The excited state exists as long as water remains in that state. Okay, and so you can think about, for example, when you have water pressurized in pipes, right? You have water running to your house in pipes, and it's pressurized, right? That's why it flows when you open it up. Now that water in that pipe, it doesn't freeze at 32 degrees. It actually has to get a bit colder because it has that pressure all around it, pushing in on it, right? And so, but when it does freeze, when it gets cold enough, it actually freezes kind of all at once because it like pushes it over this edge kind of thing. But he's talking about that in here. And another thing to understand about water, most solids, they get smaller. They take up less room than their liquids. Whereas water is different. When you put something in your freezer, in a jar, and you fill it up a little bit too much, right? We'll put some chicken broth, we'll make chicken broth and we'll put it in the freezer. And if we fill it up too much, then it cracks the jar, which is extremely annoying. Well, why is that? Because ice will take up more room than the liquid that created it, right? Just so we can put some conceptualization to what Dr. Peat is talking about here. He is getting deeply, he's in the chemistry and, difficult to understand. So probably going to take a few listens to grasp a lot of this. Okay, so you imagine being under all that water as a diver, and that's a ton of pressure, all that water above you, right? The lower you go in, the deeper you go into a body of water, the more pressure is on you. And, you've felt it before. If you've swam, into even a 10-foot pool, you've felt that pressure increase. And so with all that pressure, it's keeping what would be Viscinal water, right? That more structured water that's taking up a larger volume. But because of all that pressure, it's kind of compressing it into the smaller volume and essentially turning it into quote-unquote ordinary water, right? That bulk water that he talked about before. And that is what the excited state is, right? You hear Dr. Peat talk a lot about the excited state when he talks about like glutamate and aspartate. this quote-unquote excitatory system, and how, we kind of need to combat it a little bit with our diet, right? Because you see a lot of that in things like Parkinson's disease and some of these, neurological or nerve diseases. Okay, but as long as that bulk water remains in that state, then you're in the excited state, right? So these changes of state under pressure are reminiscent of Bose's use of pressure in some of his coherers, and of the fact that pressure alters the sensitivity of electrons in a semiconductor by altering their quote-unquote band gap, the amount of energy needed to make them enter the conductive zone. One of the early demonstrations that cell water undergoes a phase change during muscle contraction involved simply measuring the volume of an isolated muscle. With stimulation and contraction, the volume of the muscle decreases slightly, right? It enters this excitatory state, and the volume decreases. There's a phase change going from that viscinal water to that bulk water, essentially. So then he says, the muscle was immersed in water in a sealed chamber, and the volume decrease in the whole chamber was measured. This corresponds to the conversion of viscinal water to bulk-like or dielectric water. Okay, sorry. So he was getting to it. Jumped the gun. The threatening implications of those experiments with spontaneous volume change were very annoying to many biologists of my professor's generation. In the stimulated state, the cell's uptake of water from its environment coincides closely with its electrical and thermal activity, and its expulsion of water coincides with its recovery. In a small nerve fiber or near the surface of a larger fiber, these changes are very fast. In a large muscle, the uptake of water is faster than the flow of water from capillaries can match, but it will become massive if stimulation is continued for several minutes. For example, 2 minutes of stimulation can cause a muscle's overall weight to increase by 6%, but its extracellular compartment loses 4%, so the muscle cells gain much more than 6% of their weight in that short time, and there's a study in 1996. The water that is taken up by cells is taken from the blood, which becomes relatively dehydrated and thicker in the process. Okay, and this is why people say that you see these massive bodybuilders, and this is why they say a lot of that muscle bulk is edema. It's just these muscles in this excited state that have taken up an insane amount of water. Right? You can get online and watch these competitions between, I've seen one where it's like, bodybuilders versus farm guys, farmers, farm guys. Anyway, farmers, they have a name. And somehow, the farmers are beating the bodybuilders, even though they look not nearly as, ripped. And it's because it's just muscle content over here in this one category, whereas over here it's muscle content plus edema. The belief in quote-unquote semi-permeable membranes, which hasn't been a viable explanation of cell physiology for a very long time, forces people to explain cell swelling osmotically, which means that they simply assume that the number of solute particles inside the cell has drastically increased in a very short time. Okay, solute being the thing that is dissolved in it, right? If you think about a saltwater solution, you have the solvent being water, and you have the solute being the actual salt that is dissolved, right? I feel like I'm back in teaching my high school chemistry classes here. Okay, in Tasaki's experiments from 1980, 81, and 82, the swelling in a nerve coincides with the electrical action potential, which, according to the osmotic explanation, means that a very large increase in internal osmolarity happened in essentially no time. The action potential comes and goes in about 2 milliseconds. The swelling also coincides with heat production and shortening of the nerve fiber. The shrinkage of the nerve fiber after the end of the action potential may be just as rapid, and the membrane theory offers no explanation for that either. But the restoration of the unswollen state can be very prolonged, depending on conditions extrinsic to the particular muscle or cell. Troshin's survey of the theory of osmotic regulation of cell volume showed that the idea of the cell as a membrane osmometer was false, but very few biologists read his book. All right, so we kind of get the pattern of There's some dogmatic belief, and many people have come along and proved it wrong, and they just kind of get swept under the rug. Since the excited or fatigued muscle or nerve swells and gains weight, it's interesting to see what happens to their sensitivity and strength when they are exposed to hypotonic solutions that tend to promote swelling, or hypertonic solutions that help to prevent swelling. In a hypotonic solution, cells are excited. And there's from Lang et al. 1995, and here's a quote: exposure of aortic strips from guinea pigs to hypotonic extracellular fluid is followed by marked vasoconstriction. End quote. But the early excitation is followed by decreased responsiveness. And here's another study from 1984. Quote, exposure of muscle to hypotonic solutions, 70% of normal solution, produced initially a transient increase in twitch, after which twitch declined below the control level. Hypertonic solutions tend to produce relaxation in normal muscles, including the aorta. And there's a study from 1999. But when muscle function is impaired, especially in the circulatory system, as in shock, they improve contractile function. And there's a study from 1998, and here's a quote: the maximum contraction force measured in isolated right papillary muscles ex vivo was significantly greater in HSD treated than normal saline treated animals. Athletes can lose 4% of their weight by dehydration without decreasing their muscular strength. Hypothyroidism tends to cause a loss of sodium from the blood, and the hyponatremia sometimes leads to a generalized hypotonicity of the body fluids. Okay, hypothyroidism, loss of sodium, and then generalized hypotonicity, okay? And if you are interested, Dr. Brewer talks a lot about this, maybe not in such technical terms. And so if you go to the Dr. Brewer Pregnancy Diet website, I think it's .com, might not be. But anyway, he talks a lot about taking more salt during pregnancy because of the increase in fluids, right? You're increasing the amount of blood in your body by a lot. I don't know what it is exactly, but anyway, well worth a read. And Dr. Peat, he talks about Dr. Brewer's research a lot, so well worth some looking into. The thyroid hormone itself functions as an antioxidant, but much of its protective effect against cell damage is probably the result of preventing cell swelling and accelerating the removal of calcium from the cell. Swelling, like fatigue, causes intracellular calcium to increase. Okay? Intracellular, in the cell, extracellular, outside of the cell, right? I'm sure you guys all have that. Just make sure we're on the same page, but swelling will bring calcium, right? The swelling, which is, the edema, which is the excited state, right, is higher intracellular calcium. So all of these things are connected. The electrical surface charging of lipids in bulk water probably accounts for the increased lipid peroxidation that occurs in fatigue, edema, and hypothyroidism, when water loses its normal partial hydrophobicity. Increased carbon dioxide is known to decrease lipid peroxidation, and its production requires adequate thyroid function. Thyroid stimulation of oxygen consumption tends to prevent lactic acid production because it keeps the cytoplasm in a state of relative oxidation, i.e., it keeps the concentration of NAD+ hundreds of times higher than that of NADH. Okay, and again, what we talked about before, NAD+, these are the taxi cabs able to grab up people to run them through the electron transport chain. Whereas NADH, that's a cab that's already taken. It can't pick up, electrons to move them along to energy production, right? NADH is required for the conversion of pyruvate to lactate. It is also the source of reducing potential in many kinds of toxic redox cycling that generate lipid peroxides. And it maintains the sulfhydryl system, involving the balance of reduced glutathione with the sulfhydryl disulfide system of protein bonds which governs the cell's electronic state and affects its balance of hydrophobicity and hydrophilicity. All right, I might name my next daughter that, hydrophilicity. Okay, bad jokes. The harmful lipid oxidation interferes with energy production and regulatory processes and is responsible for some of the prolonged effects of fatigue, swelling, and hypothyroidism. These lingering effects of lipid oxidation are undoubtedly amplified by the presence of larger amounts of unstable polyunsaturated fats as the energy demands of the fatigued state mobilize free fatty acid from the tissues. One of the oldest tests for hypothyroidism, here we go, was the Achilles tendon reflex test, in which the rate of relaxation of the calf muscle corresponded to thyroid function. The relaxation is slow in hypothyroid people. Right? We just talked about that. Your calf muscle can't relax as fast when you are hypothyroid. And it's the same in all cells, but it's just more easily perceived in the calf muscle. Water, sodium, and calcium are more slowly expelled by the hypothyroid muscle. Exactly the same slow relaxation occurs in the hypothyroid heart muscle, contributing to congestive heart failure, because the semi-contracted heart can't receive as much blood as the normally relaxed heart. The hypothyroid blood vessels are unable to relax properly, contributing to hypertension. Hypothyroid nerves don't easily return to their energized, relaxed state, leading to insomnia, paresthesis, movement disorders, and nerves that are swollen and very susceptible to pressure damage. With aging, hypothyroidism, stress, and fatigue, the amount of estrogen in the body typically rises. Estrogen is catabolic for muscle and causes systemic edema and nerve excitation. It weakens muscle contraction in the bladder, although it lowers the threshold for stimulation of sensation and contraction. And there's a study. This is the pattern that causes people to wake up frequently to pass a small amount of urine. Progesterone has the opposite effect in the urinary bladder, raising the threshold of response, but strengthening contraction as it does in the gallbladder. Estrogen lowers stimulation threshold in the gallbladder, as it does in the brain. Part of its excitatory action might be the result of increased hypotonic tissue water, but its effects on nerve thresholds are practically instantaneous. In 1971 and 72, I gave some of the reasons for thinking that estrogen's biological effects result from its direct effects on cell water, causing it to become more like bulk or high dielectric water. For example, NMR or spin echo of estrogen-treated uterus and of the uterus from an old animal were closer to bulk water than that of a young animal. Estrogen, like fatigue or excessive oxygen, slows nerve conduction. Lactic acid production increases with fatigue, aging, hypothyroidism, estrogen excess, and other inefficient biological states. Its presence when oxygen is available indicates that something is interfering with efficient oxidative energy metabolism. Ammonia, free fatty acids, and various inflammatory cytokines are also likely to increase in the stress states. A dangerously high level of ammonia in the blood, or hyperammonemia, can be produced by exhaustive exercise, but also by hyperbaric oxygen, or a high concentration of oxygen, by high estrogen, and by hypothyroidism. It tends to be associated with an excess of lactic acid, probably because ammonia stimulates glycolysis. Excess oxygen, like hypothyroidism, is equivalent to hyperventilation in producing an abnormally low level of carbon dioxide in the blood. The Krebs cycle, during stress, is limited to the unavailability of carbon dioxide. These factors result in the waste of glucose, turning it into lactic acid, rather than carbon dioxide in energy. And if you've read much of Dr. Peat, this sounds an awful like the big C word, right? In these ways, the metabolism of fatigued muscle, or any cell under stress, is similar to tumor metabolism. Okay, I just keep jumping the gun. I'm going to let Ray Peat speak for himself here. All right, hyperammonemia disturbs excitatory processes and can cause seizures, as well as stupor. and is probably involved in mania and depression. Lithium happens to complex electronically with ammonia, and I think that accounts for some of its therapeutic effects. But carbon dioxide is the main physiological factor in the elimination of ammonia, since it combines with it to form urea. The changes in cell water in the excited/fatigued state represent an increase in the water's quote-unquote structural temperature. And that would imply that less carbon dioxide could remain dissolved during excitation. Eating sugar and using caffeine, which increases the oxidation of sugar, and there's a study from 2005, can reduce fatigue, both subjectively and objectively. Metabolically, they increase the production of carbon dioxide. Increasing sugar decreases the liberation and use of fatty acids, and by a variety of mechanisms, helps to lower the production of ammonia, lactate, and inflammatory cytokines. Lactic acid in combination with acidosis and free phospholipids can interfere with efficient cell functions. And there's a couple of studies there. Free fatty acids release tryptophan from albumin, contributing to the formation of serotonin, which increases the sense of fatigue. Aspirin and niacin help to prevent fatigue symptoms and to prevent many of the harmful systemic oxidative after effects. Both are anti-lipolytic. Aspirin uncouples mitochondria. Uncoupling of mitochondrial oxidative metabolism from ATP production helps to consume the sugar which otherwise would be diverted into lactic acid and converts it into carbon dioxide instead. Mild hypoxia, as at high altitude, suppresses lactic acid production. This is called the quote-unquote lactate paradox, and increases the amount of carbon dioxide in tissues. Aspirin and thyroid, or T3, increase uncoupling. A drug that used to be used for weight reduction, DNP, also uncouples mitochondrial metabolism, and surprisingly it has some of the beneficial effects of thyroid and aspirin. It stimulates the consumption of lactic acid and the formation of carbon dioxide. Okay, well, when it was mentioned up above, I was not expecting super good things, to be honest, but all right. The squirrel monkey, which on average weighs about 2 or 3 pounds as an adult, lives much longer than other animals of its size, usually about 20 years, as long as 27. It has an extremely high rate of oxygen consumption. This is probably the result of natural uncoupling of the mitochondria, similar to that seen in long-lived mice. Mice with 17% higher resting oxygen consumption lived 36% longer than slow-respiring mice of a related strain. And that's from Speakman et al. 2004. Living at a high altitude, people tend to eat more and stay leaner than when they live near sea level. Apparently, their mitochondria are relatively uncoupled and they have more mitochondria, which would partly account for their lower production of lactic acid during muscular exertion. Increasing thyroid activity, too, tends to increase mitochondrial mass as well as their uncoupling. Most of the things that we think of as fatigue result from disturbances of the hydration of cells, whose sensitivity, composition, and structure change according to the extent of the disturbance. The hydration is governed by the cell's quote-unquote electrical properties, which are regulated by internal metabolic processes and by systemic processes. When cellular fatigue reaches a certain point, only the interactions of all the organs can restore stable cellular structure and functions. The liver eliminates lactic acid and ammonia. The adrenals and gonads provide stabilizing steroids, and the brain alters activity and behavior in ways that can reverse most of the effects of fatigue. But when the tissues contain large amounts of polyunsaturated fats, every episode of fatigue and prolonged excitation leaves a residue of oxidative damage, and the adaptive mechanisms become progressively less effective. When the most powerful adaptive mechanisms, such as the timely synthesis of progesterone, pregnenolone, DHEA, T3, and the inhibitory transmitters, GABA and glycine, fail, then some of the primitive defense mechanisms will become chronically activated, and even sleep may fail to restore normal cellular water and metabolism. Hyperventilation often becomes a problem, making capillary leakiness worse. Water in the body occupies 3 major compartments: blood vessels, extracellular matrix, and the moist cell substance itself, and its condition in each compartment is a little different and subject to variation. There are no textbooks to use in the U.S. that treat intracellular water scientifically, and the result is that physicians are confused when they see patients with edema or with disturbances in blood volume. It rarely occurs to physicians to consider disturbances of water distribution in problems such as chronic fatigue, fibromyalgia, sleep disturbances, frequent urination, slow bladder emptying, anxiety, paresthesia, movement disorders, the tunnel syndromes, or even slow thinking. But, quote-unquote, intracellular fatigue leading to overhydration is probably the central problem in these and many other degenerative and inflammatory problems. The improvements in cell functions and water distribution that are inversely related to oxygen pressure and directly related to carbon dioxide won't be discussed in medical textbooks until they have given up the idea of membrane-regulated cells. The quote-unquote treatment for intracellular fatigue consists of normalizing thyroid and steroid metabolism and eating a diet including fruit juice, milk, some eggs or liver, and gelatin, assuring adequate calcium, potassium, sodium, and magnesium, and using supplements of niacinamide, aspirin, and carbon dioxide when necessary. Simply increasing carbon dioxide decreases lactic acid and ammonia, increases GABA, the sleeping improving nerve inhibitor, and regulates mineral and water disposition. One of the outcomes of the study of the physiology of fatigue is that it leads to a better understanding of cells in general and offers some new insights into aging, inflammation, and a variety of stress-related diseases. Whew, that was a long one. All the references at the bottom, as usual. I hope you were able to make it through that. That is a lot, and sometimes you just have to say, just tell me what I need to do, right? A lot of us are going down the rabbit hole of health in general because of health problems and wanting to help people around us. But I very much appreciate how he always has, at the very bottom, pretty much, this is what you should do to fix these problems, right? And so, regardless, I hope this helps you. I hope you can share the bits and pieces that you were able to get from this with the people you love and care about. And until next time, God bless.